Hi there guys, and welcome to another A-level biology revision video with Mr Phillips. Today I am going to be continuing with year 12 content as we move on through biomolecules. Today's topic is amino acids. So amino acids are part of proteins. You'll remember from GCSE that when you digest a protein using a protease enzyme, it becomes an amino acid. So what are the functions of proteins and therefore amino acids? Well, a lot of these you will be familiar with. It's just whether you've actually linked them all together. So amino acids, or sorry, proteins are used to make antibodies. Okay, so antibodies normally have two long and two short polypeptide chains which make up the defense in our bodies. All enzymes are globular proteins with a very specific shape in the active site. Most hormones are proteins. There are non-protein ones as well, but a lot of them are. Transport proteins, so something like hemoglobin. That hemoglobin attaches to the oxygen molecule to carry it from your lungs to the respiring tissues in your body, is a protein made of 4 polypeptides. Then we have structural proteins such as collagen, well, collagen will do, can't remember. And then we've got some transport proteins, which is stuff that you'll learn when you do the membrane structure, so like channel proteins, carrier proteins, co-transporter proteins. And then in your muscles, you've got 2 proteins called actin and myosin, which contract effectively as you when you need to move. So the basic structure of amino acid is one that you need to memorize. So I always tell students to learn the amino acid structure by learning that it's got something called an NCC backbone. And I think that always helps if you always remember that and build the rest of the structure around the backbone. So effectively what you've got around the NCC backbone is the middle carbon is called the alpha carbon, okay, and then the attached in the middle to the alpha carbon is 2 molecules. There's always a hydrogen, which could be above or below it, doesn't matter which orientation it is, and then there's something called the R group. And the R group, or the residual group, is one of 20 different groups because there are 20 different amino acids. So that in the most simple case is just a hydrogen, but in other cases it's lots of different molecules. We then have something called the amine group, sometimes called the amino group, and basically the amine group is this bit here, this NH2, and whenever you're talking about that part of the amino acid, you need to refer to it as the amine group. And then on the other side, we've got the C double bond O, and then the OH, and that gives us the what I call the carboxylic acid group. So if you think about it, you can actually call the amine group the amino group, but effectively you've got an amine group acid group, amino acid, okay, and that therefore gives you a total structure which you need to memorize for the exam. So when we then look at protein, how proteins are built, obviously amino acids are the monomers of proteins. So here again we've got the basic structure of amino acid. One that I would suggest that you do learn though is the structure of glycine, and the reason glycine is so important is because the R group is a hydrogen, so it is the smallest amino acid, and it's often used in collagen, which is a structural protein, and basically because it's got the smallest R group, the chains of collagen can fit really close together, making it more stronger and structurally useful. So amino acids then join, basically giving a repeated backbone as NCC, as I said before, and when they join, we're going to look at this in a bit more detail, they will form a long polypeptide chain like this, but we're going to look at how they actually combine in the next slide or so. And the last thing just to point out about the R groups, so we said in the last slide there are 20 different R groups. Well, the R groups can have different properties depending on the atoms in them. So for example, they might be positively charged, might be negatively charged, might be hydrophilic, which means they're attracted to water, or they might be hydrophobic, which means they're repelled from water. So that obviously has a big effect on the amino acid, and then overall that will have an effect on the shape of the protein molecule. So there are 20 different amino acids, as we said, just to give you some extra information. Plants can make all the amino acids they need as long as you have the essential nitrogen in the soil, so the nitrate in the soil to make that NCC backbone. And this can be turned into the amino acid group and onto amino group, the amine group, sorry, and then they can join the products of photosynthesis and can be attached to different molecules to make amino acids. Animals, however, do need amino acids in their diet, okay, and we obviously break down proteins and then we rebuild the amino acids to make our new proteins. We can convert one amino acid type into another amino acid type in a process called transamination, but that we can only do that for what we call non-essential amino acids. So what I mean by those is we can make certain amino acids in our bodies and they're non-essential for our diet. However, there's about, I believe, 8 amino acids that are absolutely essential for our diet because we are not able to construct them ourselves using transamination. So they are essential parts of our diet. We must have them in our diet. Plants contain fewer amino acids, so people who are vegetarian and I presume vegans as well need to carefully balance their diets. One thing that does contain all the essential amino acids is soya, which is obviously a very popular choice to ensure that the vegetarians and vegans have that all those essential amino acids. So again, joining the amino acids together, if we have a look at this, it's not a particularly different molecule from the one we would have seen before. So what we've got effectively here is individual amino acids join up. So we've got 2 separate amino acids here at the top. So we've got 1 amino acid there and a second amino acid here, and they're going to join up to make a dipeptide molecule. So dipeptide molecule just means 2 amino acids, and the word peptide is strongly associated with amino acids. So let's have a look at what happens then. So basically we've got, and you need to be trying to remember to refer to the names of the groups. So if you remember over here, we've got the carboxylic acid group of this amino acid, and then here we've got the amine group or the amino group of another amino acid. So when you refer to this in the exam, don't just say the OH and the H bind. You need to say about the carboxylic group, which is one I'm just deleting the circle from now, and then the amine group, which is there. Now in terms of what happens then, exactly the same as we've talked about with sugars, but apart from that there's a different enzyme, so there isn't going to be, and the different molecules are involved, so there isn't the need for the oxygen to form the bridge. So at this point, what we've got is 2 molecules of, 2 different molecules of amino acids, and the OH from the carboxylic group and the hydrogen from the amine group combine. That means a water molecule can leave. Now what that leaves us with at the top, so if we imagine all this is gone now, what that leaves us with is some molecules with interesting valencies that we can use. So we've now got a carbon atom here, okay, and this carbon atom has got valency of 4, so it's using 1, 2 up to that oxygen, it's using 1 up back to that carbon atom, but this one no longer combines down to that OH. So that one can now just go straight across there. And if you look at this one, this nitrogen, which has a valency of 3, that one's still going to carbon, that one's still going to the hydrogen, but that nitrogen bond can then join up with the carbon and use its final valency up there because this one is no longer here. So if you look at it, the water molecule has come out, so it is a condensation reaction, as you will remember, to form a covalent bond, but the covalent bond is formed. It can never be called a covalent bond in the exam because they will want another specific name, and the specific name is a peptide bond. Okay, so the peptide bond written over there. So you can see now we've got this repeating NCC backbone, NCC backbone, and this is where your original amino acid was in 2. So we can just see now that the OH has gone from this carbon, okay, the hydrogen has gone from this carbon, and they've joined in the middle like that, making a dipeptide molecule. Now when the when we want to break dipeptide molecules up, and you'll learn about something called dipeptidases later in the course, we do the opposite reaction again. So if you remember, this would be a hydrolysis reaction. And a hydrolysis reaction is where we put a water molecule back in, okay? And by putting the water molecule back in, we effectively get rid of break the bond between, break the peptide bond between the amino acid dipeptide structure, and that takes us back up to this situation here. And obviously a hydrolysis reaction is using water to break the bond. So also you need to just remember from GCSE really the protein test. So the protein test for proteins is called the Boyer test, and the Boyer test we use sodium hydroxide and a few drops of copper sulfate, and basically the reaction reacts with the peptide bonds in the protein, and that goes from a blue color, which makes some people get confused with Benedict's test, but it isn't. It's definitely Boyer test, and that goes to a purpley colour if there is a positive result. Now, I think it's always important here. A lot of people get Benedict's and Boyer confused. So if you think of it as being a protein, we don't like heating proteins because they denature. So this method does not involve heating, and it will convert rapidly to a mauve or a lilac or a purple colour as soon as you get a positive result. Last few bits then, amino acids can act as buffers. So buffers are things that can resist slight changes in pH. So amino acids can act as bases when they're put with in acids, and the reason they'll act as a base is because the COO group will happily gain a hydrogen. So you see on this initial one there's no hydrogen, then it will gain a hydrogen to become a base. They can also act as acids because they will lose protons in order to act as acids as well. Amino acids can be poison, so you learn about this in at year 13, and basically the poison side of it is that amino acid, if we eat too much of anything, it can be toxic, but if we have surplus amino acids, so more amino acids than we need, it can build up to be toxic. So the liver is what breaks excess amino acids down in deamination, and this is a two-stage reaction where the excess amino acid is converted first into ammonia and then into urea, and then the urea leaves the liver, goes through the blood to the kidney, and the kidney excretes urea to the bladder with a mixture of water and salt, which is known as urine. So I hope that's been useful for you, and you understand the basic properties of it. Really important to make sure you remember the complete structure of amino acid. You only need to know the R group. You don't need to know different examples other than maybe glycine, which has an R group of hydrogen. So thank you very much, and I'll see you next time.